So guys, I'm proud to introduce Paul Dean from the Department of Psychology at the University of Sheffield. Paul Dean studied as an undergraduate at the Trinity College of Cambridge. You can't hear me? Come on. He was a research student at the University of Oxford with Flyway. Is it better, you think?
Yeah, sure.
He supervised about REALNET, then he moved to the University of Sheffield to work on the superior curriculum, but later became interested in modeling, first of the oculomotor system, then of the cerebellum. He's currently collaborating with Professor Efferent in the REALNET project, and he's going to give us a nice talk about adaptive filter models of the cerebellum, spectral fiction.
Okay, first of all, actually, I would like to thank Professor Efferent for inviting me here, and also for the members of his group who've been very generous in sharing their time, and in particular Avi, who has been a truly excellent guide. So thank you very much. Okay, so I want to talk about adaptive filter models of the cerebellum. So now they're almost 30 years old. This was first put forward by Fujita in 1982. And even that is based on older ideas, more Albus ideas, which are about 10 years beforehand. And so we were quite interested to find that when we carried out a recent review of cerebellum models that are concerned with control, so typically eye movements or arm movements, that many of them seem to share features with the adaptive filter model. And so the first part of my talk, I want to address the question, well, why are they so popular? And just to start with what the adaptive filter model has in it. Okay, we start, sorry if this is a bit simple, but just to start with a fixed filter. Okay, so here we have, in panel B here, we have some device that transmits low frequencies and essentially blocks high frequencies, a low pass filter. We can plot its gain against frequency in a Bode plot, and we can also describe its performance as a response to essentially an impulse, its impulse response, which in this case is something looking like that, an exponential decaying response. And we're familiar with these in, for example, audio systems, where actually we can adjust them. We have knobs which enable us to which enable us to increase the gain, lower the gain, or alter the frequency response. And in some cases, these can be really quite fancy. So you can have a graphic equalizer where we can actually adjust individual frequency bands so we can get just the kind of tone that we want. But these filters still have to be adjusted by us, have to be adjusted by hand. And actually, what we want is something that does what we want it to do automatically without our fiddling with knobs. And what we need is some sort of adjuster signal that tells the filter what it is we want to do. And the version that seems to be mainly in use is something called the analysis synthesis filter. And it's called that because there it is. So we have our input here, illustrated by a simple impulse. And the first stage of the filter, it analyzes it's a whole bunch of different versions of itself. Now, in this particular case, they're leak integrators with different time constants, but there's lots and lots of different ways of doing it. But the point is that each of these different components is weighted, like so, and recombined or synthesized to produce the outputs. And the idea, since these weights can be positive or negative, you can sculpt any impulse response that you like by the correct selection of the weights. And the thing is that these weights, their values are adjusted by some external signal in the system. Just to avoid future confusion, I'm using the term adjuster signal here. That it's sometimes called a teaching signal, sometimes an error signal, sometimes a target signal. I may well be using these terms indiscriminately, except at a certain point. But anyway, for here, it's an adjuster signal that is going to adjust these weights. And the learning rule is the following one. So we have our 1 to n components, and we have our teaching signal, and for each component, we ask, is there a positive correlation between the signal on this component and the adjuster signal? And if there is, we reduce the weight. If the correlation is negative, we increase it. And so learning will stop, the weights will no longer change, when there is no longer a correlation between that component and the teaching signal. So in fact, this filter implements a decorrelation algorithm. So I now wish to make 3 points about why this device is popular as a model of the cerebellar cortex. First of all, is they work. They're actually computationally very powerful and apparently very widely used in signal processing, and even financial engineering. So that's the first point. It gives you power. However, it still doesn't mean it has any link with the cerebellum, and so there's a couple of things to say about that. First of all, it's thought by some that there is a resemblance between the structure of the adaptive filter and the basic structure of the cerebellum. So that's another version of the analysis synthesis filter in panel B. In A, it's kind of colour. Why does that keep disappearing? Oh, there. What am I doing now? Right, so the input signal comes in through the mossy fibre. This bank of fixed filters correspond to granule cells. The wakes correspond to the synapses between the axons of the granule cells, the parallel fibres, and the dendritic tree of a Purkinje cell. And the weights are adjusted by the climbing fibre input. Okay, by the climbing fibre input to the Purkinje cell. And so people have seen this as there's some kind of similarity between the two structures. And the idea is that, rather than the word go, this model explains two very striking features of the cerebellar microcircuit. The first is that granule cells, there's an enormous number of research, about 80% of the neurons in the brain are granule cells. And the idea here is that you are going to need a very large number of fixed filters at the analysis stage to make sure you cover all the bases. In biology, we don't know what's going to happen. You've got to be prepared for anything. And especially, in fact, if we're dealing with nonlinear problems, then we're going to need very, very large numbers of these filters. And the second feature is the somewhat unusual behavior of the climbing fiber input to the Purkinje cell. So the idea is that what we want and what we need from an adjuster signal is that it's got to affect all the weights on the Purkinje cell. So in some sense, it's got to be extremely powerful. But we don't want it interfering with the output. We don't want the adjuster signal coming from it. That's not what we want. And so the idea is that this fits with what climbing fibers do, that they fire at rather low frequency, but they have a massive synaptic presence in the dendritic tree. And so the suggestion, yeah?
Why does the adjuster signal not affect all weights? Why could it be weight specific?
Ah, right, okay. The idea is that what you're trying to do is produce a single output. Okay, and what you're going to do is use the difference between that output and what the desired output is. So you just have one error signal, and it's got to apply equally to every single component. Okay.
But you can measure the correlation between this error signal and each input separately.
Why would you want to do that? Why would you want to do that?
The correlation bit.
Yeah, but, I mean, you're doing, effectively, all you'd be doing if you did it separately. You might as well do it. Yeah, okay. Right. Okay, so let me say straight away, as Professor Yaron's guest, not everybody believes. Okay, the third point is a link with, I put quote cerebellar functions because it's a bit of a changing concept, what characteristics of cerebellar functions. But as far as I can judge, the adaptive filter really is specially suited to carry out two of the functions that are most associated with the cerebellum. First of all, the prediction of the sensory effects of one's own movement, and second, actually accurate movement control. And I'm going to illustrate these with respect to a particular biological control problem, which concerns reafference. And the reafference problem is that your own movement affects the sensory inputs that you get. So here's a little example which will illustrate how just when we walk, the retinal image is going to pop up and down. And what this means is that some of our sensory signals, if you like, are contaminated. Okay, they've got interference with them, produced by you. And you must have, as animals, you must have some algorithm for removing this. Now, one obvious suggestion, well, actually it was obvious now, I don't think it was obvious when it was first made by Helmholtz in the 19th century, is that what you do know is what you asked your body to do. So, for example, you know I've asked myself to walk. And so I have at my disposal a copy of those motor commands, which maybe I can use that to estimate what the interference is. But how's that going to work? And actually, I think there's a clue from, this is again from non-biological signal processing, a book by Widrow and Stearns. They proposed a noise cancellation architecture, which is actually suitable for a very wide range of problems, including, I think, the noise cancellation algorithm in an adaptive filter. And we thought we'd test this out, as one would, on a robot rat. So this is part of the BioTact project, the European Framework project. And the rat, well, that's meant to be a rat. I actually don't think it looks very realistic, but funny enough, when it, hang on, what's going on here? Sorry. Get this. But once it starts to move, and you can see actually it'll orient to the ruler. There it goes. Gotcha. Right. And the problem with this robot rat is an example of the problem I was talking about earlier, which applies to us, is that the idea is that actually this is going to be used possibly in underground dark situations to explore environments where it would be dangerous to send a human. But to explore, it needs to move its whiskers. It's called whisking. Rats do it all the time. But the problem is, mechanically, what that does is produce these spurious signals in the whiskers. And actually, I don't think there's an example there, but it will ghost orient. It will be whisking around something and whoop, and there's nothing there. And so actually, our role in this was to try and find a way of getting rid of that. And so what we used is the noise cancellation architecture in the reafferent problem structure. And so, okay, so the first part of this is, here's the motor command to the whiskers. It passes through all the mechanics of the whisker joint and the motors that are used to move it, the plant dynamics. That produces this signal, and the term that's sometimes used is reafferent signals, meaning it comes from me. Now, it's necessarily mixed with any external signal, including something we want to orient to. It's mixed together, and you've got this contaminated observed signal. And the idea is you send a copy of this motor command to our adaptive filter. And what we're going to try and do is to get this filter to predict what this signal is going to be. Because in principle it can do that, provided it can learn these plant dynamics. And so what we then do is we have some comparator structure which subtracts this signal from the observed signal, and that's now our estimate of what the external signal actually was. And so you can see if this was a perfect model of the plant dynamics, then this would be a completely accurate estimate of that signal. Now, I think, in some ways, I think actually rather the nice thing about this is that you can then use this estimate as your adjuster signal. And the logic is this. If there's a correlation between the motor command and this estimate, we know there's still contamination there, because it's coming from us. If what we're doing is influencing that, somehow we've not got the right prediction there. So if we can genuinely decorrelate the adjuster signal from the input signal, then we will have an estimate which no longer contains this information. So in other words, the decorrelation algorithm is beautifully suited to this kind of problem. And the thing is, actually, this is where the terminology may begin to matter. It doesn't require any kind of non-biological deus ex machina telling you what you need to do. It's intrinsic in the information available to you. Okay, so does the algorithm work? Well, I guess you've guessed by now. Yes, it worked actually not too badly at all. The work was done by my colleague Sean Anderson and the Bristol Robotics Laboratory, and they now incorporate it as standard in their whisking robots. Relevance to biology.
Can I ask a question?
Yeah.
It's often the case that there is correlation between movement and the sensory signal. External, for example, if I look upstairs, if I look up, I see the sky. If I look down, I see the ground. The sensory input is not the same.
Yeah. This is a particular subset of active sensing problem. For some problems, you actually need to deliberately correlate to in order to estimate the inputs. But for this particular purpose, we just want to get rid of it. Okay, the relevance, or perhaps I should say to biology, to cerebellum. It's actually relatively recently been suggested that the cerebellum is involved in learning sensory predictions. Previously, the heavy emphasis was on motor control, but now there's more consideration of sensory prediction. And one of the examples from the 90s is that tickling yourself is not easy. And the argument is that's because you know in advance what it's going to feel like. Because of the Efferent solution, that we do in fact, like the rat, have a model of our internal structure, and so if we try and tickle ourselves, it doesn't work because we're subtracting out the predicted information. And the idea of some imaging evidence is that this attenuating signal comes from the cerebellum. And just a final point on this, more generally, that again now a very fashionable approach to thinking about cerebellar function is to talk in terms of model control, internal models for control. And this is actually an example of a forward model because it takes the forward command through the plant to get the sensory output, and it makes a forward model of this plant. This filter is meant to model that plant. And that's considered apparently to be a very important fundamental cerebellar function. Okay. So, I'm saying here that, or I wish to argue that, the adaptive filter is specially suited to carry out two functions. The second one is accurate movement control. Because up to now, I've only talked about sensory effects. And I want to take as a kind of thought experiment what we've been doing so far. All the comparisons have been taking place inside our heads. And how would that work if we wanted to get rid of this interference? As we walk, our retinal image is doing this. And of course, we're not aware of it. We do get rid of this interference. How would we do that? And what I term here the sensory solution is the sensory noise cancellation framework. Okay, so what we have here is we have head motion, and what that does is effectively wiggle the eyes, and it produces image movement, which is often referred to as retinal slip. You get slip of the whole image over the retina. could use a signal that tells us about head motion, which could be, again, an efferent copy, a motor command, or it could actually come from the canals themselves, because they signal head movement. And we could learn a model of this process, and we could use the adaptive filter to learn an estimate of the retinal slip that is not produced by head movement, which actually is usually zero. I mean, in this particular case, there is normally no external source of image movement. But there's a real problem with that. It still means the image is smearing over the retina, and that messes up visual processing. Irretrievable. You can't get it back. Your acuity is done. And so, wouldn't it be better if we could stop that image movement by moving the eyes to counter the head's movement, to maintain the image? And image stabilization seems to be extraordinarily important for this. This is actually, of course, not quite the same. The animal's holding its head still while the body moves. Can you dance? Okay. Stop it. So, in other words, there are times when we don't want a sensory solution to the motor cancellation, as the sensory solution to the noise cancellation problem. We want a motor solution, and this, the one that we use mainly, is to counter-rotate our eyes. Yes, so this is something that ocular motor people use. Suggest if you've got a piece of paper in front of you, you can try jiggling it and seeing how well you read it, and then try and jiggle your head, same frequency, and see how well you read that. And with a bit of luck, you'll find it easier when you jiggle your head. Anyway, so the noise cancellation is now done in the external world. It's not inside the brain, it's in the world outside. And that does mean you need some changes to the architecture. The first one here is that the output of our filter actually now is a motor command. It's not an internal signal, it's now controlling some muscles. In fact, in the case of the VOR, the basics are done outside the cerebellum altogether. They're done through the brainstem. And what the cerebellum does is boost it, if you like it, fine-tune it. And so it sounds slightly odd, but the filter now provides an estimate of the additional eye movement commands you need to avoid the effects of head movement. So it's an estimate of the command, not an estimate of the sensory signal. The comparison is now taking place not inside the head, but actually in the retina. And the signal for changing the adaptive filter is now whether or not there is retinal slip. As I say, normally we can regard this as zero for this reflex. I think that's fair. And the third change is you need to have a copy of these commands sent to the cerebellum. And the reason is that the oculomotor plant is a term used to refer to the mechanical properties of the eye muscles and the cells and the tissues in the orbit. And the thing is, this can be regarded as a source, if you like, potential interference with your sensory signal. Because supposing the plant changes, supposing your eye muscle weakens, or when you grow up it gets stronger, if you just send the same commands, you're not going to produce the right movement of the eyes, and so you'll get retinal slip. So what you need to know to avoid that is you need to have an Efferent's copy of the motor commands, which we refer to as a recurrent architecture. But the logic is still the same, actually. It's the decorrelation principle that if there's a correlation between motor command and sensory error, then we assume it's causal and that the motor command is incorrect. And so we need to remove the correlation between motor command and its sensory consequences, these particular sensory consequences. And this seems to work well in simulation. And for those who are interested, it's relevant to a particular problem, I think, in adaptive control theory, which is the distal error problem, where actually you don't know what you should be doing. But I just mentioned that as an aside. Again, working with the Bristol Robotics Laboratory, we've used this architecture to stabilize this robot head thing, camera, stabilizer camera. Okay. So, a couple of summaries. First, I'm arguing that the basic sensory prediction architecture can be altered so that the sensory noise caused by the movement can be avoided altogether rather than simply predicted. I'm suggesting that this can form the basis of a method of calibrating movement accuracy. So, for example, if your saccade is inaccurate, its target image will be off the fovea. And you can use this in a similar method of calibrating accurate saccades. And finally, we wish to argue that the decorrelation algorithm can be used for motor control in a biologically plausible manner. And final summary of this section. Okay, so the first part of the talk, why are people finding these filters attractive or plausible? Those are the three reasons. They're computationally very powerful, they appear to have the same basic structure as cerebellar cortex, and their operating principle fits very well with two major basic cerebellar functions. But the key thing is that may very well be the case, but is it true? And so the second part of the talk is addressing the issue, well, how might we find out? And so I'm going to, in this sense, treat the adaptive filter model as a hypothesis. And a quote from David Robinson, who I think spent much of his time trying to convince experimentalists that they should listen to modelers, is that models are simply hypotheses. To be against models is to be against hypotheses, which in science is like being against motherhood and apple pie. So, how can we test it? Right, now the framework that we're thinking about at the moment, a good way of testing it, takes two critical features of cerebellum. First of all, the microcircuit itself is broadly, if you look at it under a microscope, it's not easy to tell which bit of the cerebellum you're looking at. But each individual region of the cerebellum has its own unique connectivity. So there's this odd mixture of uniformity and difference. And so, okay, so this is a sort of cartoon from a textbook. And what it shows here is the origin of the climbing fiber signal is a structure called the inferior, actually it's easier if I do it with my finger, the inferior olive down here. That's the origin of the climbing fiber signal. And the idea is if we take a little cluster of neurons in one part of the inferior olive and follow their projections, they fan out across a parasagittal zone, or cerebellar cortex like that, and another one forms a different zone, like so, I guess just how does this fit in with what I was saying earlier, that the Purkinje cells, which one have we got that way around, and the parallel fibres are coming at right angles to them. But the idea is that we can define parasagittal zones of the cortex by reference to their olivary connections. And this is a particular very simplified diagram. You're looking at the rat cerebellum, just the posterior half, and what they've chosen to do is colour you can see the different coloured stripes there, so that if we take this one, this sort of pale green one here, that's zone A, or the vermis, it gets its input from a particular region of inferior olive, and also incidentally projects to a particular region of the output structure of the cerebellum, which is the deep cerebellar nuclei. And in fact, people have suggested that you can divide up these zones rather further, that there's even more detailed microorganisation within a zone. And it's been estimated there's something like I think this is a very crude number, but something of the order of 5,000 functional units in the cerebellum. And one way people have thought about this is you can think that a bit of cerebellar microcircuit, you can think of it as a sort of plug-in chip. And in each of these microzones, it's plugged into a particular muscle fibre input and a particular timing fibre teaching signal, and it plugs into a particular part of the deep cerebellar nuclei. So you can imagine a series of banks of these chips, all in science fiction case, all connected to different parts of the central nervous system. Okay, but now the point of talking about that is that it suggests there's two different ways we could test the adaptive filter hypothesis. First of all, is it actually compatible with the operation of the microcircuit itself inside the chip? And the other is, can it explain the connectivity of individual chips? And I'll start with the latter. And I'm going to start, I think it's all downhill after this bit, from the adaptive filter model's point of view, but I think as far as image stabilisation and the flocculus is concerned, that the evidence isn't that bad in favour of the adaptive filter. Okay, so the flocculus is a region posterior and lateral here, and I think it's generally agreed that it's involved in image stabilisation. There's classic inactivation and lesion studies and more recent studies of mutant mice. So it's involved in image stabilisation. It gets the right mossy fibre inputs. It gets a vestibular input which tells it about head motion, and it receives a massive efferent copy input of eye movement commands. Exactly as the model architecture requires. The putative adjuster error signal, the climbing fibre input to the flocculus carries information about retinal slip. That's not all it carries, but it does carry that information. And its output, floccular Purkinje cells project to ocular motor neurons, sometimes just bisynaptically in some cases. And there have been it's been a topic of considerable interest, so there's something like over 13 vivo studies in primates alone, that their firing rates can be related to eye movement. And there's been detailed quantitative analyses of firing rates in relation to variables such as eye position, velocity, and acceleration. And finally, does PC firing change in the way predicted by the adaptive filter model? what happens when you artificially change retinal slip. Now, this is a really complex issue. There have been many, many experiments done on it. And there's various reasons for that complexity, not least is that you get plasticity into the brainstem where these neurons project. But I think I would like to argue that, in general, it's broadly in agreement with what we would expect from an adaptive filter. So, I think I would like to argue that the cerebellum can behave as an adaptive filter, but that's a quite different question from whether it does so generally. And this is where we run into a problem. And the problem is this, that there are very few of those 5,000 microzones where we know the task in which it's engaged and the connectivity which it does it. And in particular, we don't know how the microzone output affects behavior and thus the timing fiber input. We can't close that loop. So that's really important in the case of cerebellum, is we know what we're dealing with. We're dealing with the flocculus alters eye movements, and eye movements alter retinal slip, and they all do it in the right direction. But for many other regions, we simply don't know this.
So for the eye movement, it's fine?
Yes, I think so. I think we do know it in this particular case for the flocculus. But I'm saying that, unfortunately, for most regions, we simply do not have enough detailed knowledge of the connectivity.
But the question was,
Yeah.
it can be doing it. Does it do it?
Sorry.
So
Let me be clear. Can the cerebellum behave like an adaptive filter? And I'm saying, in this particular,
it can.
It can.
But you asked whether it does.
And I think it does in the flocculus.
But what is the evidence that it does in the flocculus?
Okay, so the evidence would be the connectivity, the nature of the signals within it, the lesion evidence, and what happens when you do the plasticity.
What is the connectivity?
Right.
Internal connectivity in cerebellum?
No. So let me just see if I can pop back here a bit. It's working well. Okay, so these slides were summarising, very briefly, I understand, but summarising the evidence that we know the flocculus is involved in this task, image stabilisation. We know that it gets the inputs that you require from the architecture for image stabilisation. We know it gets the right timing fiber input. And we know that the commands it sends out are directly.
Fine. So
okay.
The next thing would be to do perturbation. Right? Otherwise, it still doesn't get consistency out of it. You really want to show that if you perturb the flocculus in the way that the model predicts, you see maybe behavioral changes.
Well, yes, okay, so I'm sorry I covered that very briefly, but that would be perhaps part of the inactivation studies where you manipulate,
Okay.
or you stimulate it.
And this has been done.
Yeah. Sorry, it's a very brief summary of a large number of studies. Okay, and so I'm saying in the case of the flocculus, we can actually test the hypothesis because we know this loop. We know how it should work. But
what is the gap in other modalities?
Right.
You mentioned that there is a gap in the loop. What line is missing?
Right. Okay. So just a minute. Hang on. Okay. For many of them, it's actually not. Probably, you may know something about these inputs. You very rarely seem to have a clear idea of what these are doing. So that the output's going into the brainstem, and then what does it do? You don't know. It goes to, say, a structure called the red nucleus. Where does that go? We don't know the anatomical details or the physiological details, and consequently we can't say. Supposing these cells started firing more, what's going to happen to your behaviour? Well, often that's not known. And then even if you do know something about that, how is that going to influence this signal back that you're getting on the climbing phone? You don't know that either. And I think it's not I'm talking about a deep mystery here. It's just that there's an awful lot of basic anatomy and electrophysiology that still needs to be done before we can even test whether this is a good news thing to do. I think I would argue that there are some exceptions to that. I think eye blink conditioning we perhaps know enough about the circuitry, and making accurate saccadic movements. But even if that's the case, even if we accept that, I'm saying something like, okay, this is crudely drawn on, where I think we do know what's going on. The rest of it I don't think we do. It's unknown territory. Something like the information...
What about the VOR?
Pardon?
What about the VOR?
Yes, so that's... Sorry, the query there refers to... I'm not exactly sure where the flocculus is in it, because they don't label it, and I've had to try and guess. Sorry. The flocculus is in a zone of its own. So that's the first test. The second test is the model consistent with what we know about the cerebellar microcircuit? Now, if you remember, the initial claim is it's broadly consistent with some major features of the cerebellar microcircuit. But what about other features with which it's not consistent? And the one, this is not the only one, it's one of particular relevance to this group, is the issue of Purkinje cell bistability, which is that in many circumstances it appears that Purkinje cells, their firing is interrupted by periods where they don't fire, down states and up states.
When the enemy is sleeping.
Sorry?
When the enemy is sleeping.
Well, the time scale here is, well, so this is 400 milliseconds, so it's the order between half a second and a second that it's occurring.
No.
Yeah?
I'm not sure that this causes happening in the behaving enemy.
Interesting. You should.
No, it means
Right. Okay. I think maybe this will be coming up.
The enemy.
But the point is that these down states with no simple spikes, they simply look to be incompatible with the adaptive filter hypothesis. Now, I think the first point I want to make about that is, okay, is it a case that this is some sort of artifact, or is it that bistability plays a major role in task-related firing flocculus? That is to say, when the animal is it's being rotated on the table and it's constantly moving its eyes to stabilise the image. What happens when you record from Purkinje cells in the flocculus during that? And basically, I think the idea is you don't see it. That's my understanding. The flocculus in behaving animals, when you're asking them to do the VOR, you don't see these long down states. So, is it an artifact? I think it's generally agreed that it's easier to see in slices and in anaesthetized preparation. But there is at least one study describing long pauses in awake animals, and that's done quite recently, 2009, by Yartsev et al. Pauses mean duration 700 milliseconds, so long. Whereabouts were they? Well, one is in zone A, vermal lobule. CRUS2 probably C2 or C3, I would think, localization region. What was the relation to behavior? Well, in this particular study, it's not easy to say because basically the animals were trained to be quiet. And they comment that there were no obvious behavioral correlates. So there's an issue. What's the functional significance of bistability in these regions? But what about more lateral regions? And the reason for raising that is that if we go out far enough into sort of blue zone, D0, laterally on the hemispheres, that region receives from the principal olive, the region of the inferior olive, the principal olive. And in contrast, the flocculus, which actually isn't shown on this, it receives from a different region, the dorsal cap of Cui. And there's been a study done by Rodolfo Linus's group that the neurons in these two regions of inferior olive differ. And in particular, the neurons that project to the lateral hemispheres show subthreshold oscillations and robust rhythmicity in the 6 to 12 Hz region. DCK neurons do not show this. And they argue that this may be related to differences in the functionality, that ocular motor control, you don't want tremor. You don't want your eyes flicking about. It's a really bad idea. But sensory motor control, you do have physiological tremor. And so the suggestion is maybe what we are looking at here is an adaptive filter mechanism for the flocculus, but something else going on in more lateral regions. And why might we think that? Because of evidence that the switch between up and down states can be triggered by complex spikes. And it's possible that neurons in the dorsal cap of Cui that project to the flocculus do not regularly trigger these switches, but those the principal olive do. And this has been suggested could be the basis of a hitherto not understood mode of motor control. Connected in some way with the multi-joint organisation of us. It's so complex. We need a new mode of motor control. This would in turn need a new role for olive input, not just as an adjuster signal, but as a direct driving of motor output, generating temporal patterns which control the muscles. This switching between states of Purkinje cells could be very important for such a role, and I think it could be consistent with the idea of evolution of cerebellar function from the early mode, adaptive filter, because the vestibulocerebellum is the oldest part of the cerebellum, to a new computational mode in the lateral hemisphere. So, finally then, I think this possibility raises important questions. If there is a new computational mode for the cerebellum microcircuit in lateral regions, how does it work? I'll explain how the adaptive filter works. How does this work? Can we use it to control, for example, a multi-joint arm? Does this new mode simply replace the adaptive filter, or in some sense complement it? And if so, how? And what happens, of course, in these other microzones between the flocculus and the lateral hemispheres? And so I think the summary is trying to test the adaptive filter hypothesis, given the adaptive filter is such a plausible model, that the connectivity or input-output test, where the information is available, it seems to be consistent, but as yet there is a very small number of microzones. In the microcircuit test, there are some microcircuit features which seem actually really compatible. They seem very compatible with the adaptive filter, but clearly there are others that do not. And I think it's a recurring theme. The bistability is one such feature, but there are others. Does it reflect a new computational mode of cerebellar function, or the fact that we don't have a good understanding of how an abstract signal processing model like the adaptive filter model, it has to be implemented by neurons which are slow, spiking, noisy, prone to die. Is it going to reflect some aspect of the implementation rather than the computation? And I think the future, of course we need more experts, but possibly also more modeling, because if what I'm saying is correct, we really need to construct descriptive, detailed models that are compatible with adaptive filter model function, so we can see whether a given feature really does fit or not. And this is part of the purpose of an EU-funded REALNET project, which include the Sheffield and Jerusalem groups. And this actually, it turns out, is an example of a very broad current physiological framework that it's important to model a physiological entity at different levels of description. Okay, so if I could just thank my, draw attention to my collaborators. All the maths was done by my colleague John Porrell, mathematician and theoretical physicist, who worked with robots, was done by Sean Anderson, a control engineer, and in conjunction with the Bristol Robotics Laboratory. Thanks very much.
But it seems to me from the description is that for most of parts of the cerebellum we don't actually know its effects on behavior. So what's the point of doing multi-stage modeling?
If we could, okay, we have to get the microcircuit right. We have to understand the algorithm. And the modeling is going to help with identifying that. The work in finding out which different bits of the cerebellum connect up has also got to be suited. So that at least, I mean, even if we knew for sure it was an adaptive filter, we would still have this issue of having to work out how it connects, say, for arm reaching or in language. So I think there's just two research streams which can be pursued, with both of which can be pursued.
So you distinguish between two functions of the cerebellum as one sensory and one making accurate movements. And I think if you look at your skin and also the skins you make reference to, it's not two, it's one. Because you make accurate movement if your prediction of the sensory consequences is correct. So it's all one loop in which you decide that there is two purposes. One, to stabilize the images, and one, to stabilize the movement. But it's one consequence of the other. They are related to each other directly.
Yes, I mean, I think okay, so I think the argument is that there are actually two ways of getting rid of interference produced by your movements. And one of them is inside the head and involves memory, and the other involves memory. And so, for example, you might, okay, people argue that when we make a saccade, when we make a fast movement, that what happens there is that's a deliberate movement you're trying to make. So you can't, in some sense, null it off. So when it's the VOR, the idea is that you're trying to keep the gaze still, and there's external forces preventing you from doing it. So if, in fact, your body or head moves, then you move your eyes to counteract that. But now supposing I want actually to move my eyes. I want to stop looking at you and look over there. Now I can't do that. I have to put up with those sensory consequences because it is a voluntary movement. Right? So the VOR is response to if you like, a response to people messing me about. Here I'm doing the messing about, and I can't avoid it. And so the suggestion is then that the system takes some kind of measurement of the nature of the saccade and readjusts receptive fields in the regular cortex. And that, so in other words, does that make sense?
Yeah, I don't think I completely agree to this, but it makes sense.
Yossi, do you have any questions?
Sure. Well, it seems that you made a lot of effort to keep everyone happy. And this is an admirable goal, and all the people will be. But the question is, to claim there is a change in the function of the cerebellum over evolution, while the basic circuit, you agree, is the same. The climbing fibers are still there, and they're supposed to adjust your adaptive filter. And the parallel fibers are still there, and they're supposed to fire. Everything is there.
Yeah.
So, really, it's difficult to buy that there is a change in function or a change in operation of the system.
Well, I know what you mean. But the thing is, there is that Urbano paper. Actually, there is a little bit more than that, because the flocculus is slightly odd in other ways. So, but all I'm saying is that I think it's easy for somebody from my perspective to say, oh no, it must all be an adaptive filter.
But what is left from the adaptive filter thing in the new area of the cerebellum?
It's not clear. I mean, I think that's one of the things this project is going to be about. Is there two different modes in different times? That sometimes you use bistability and Olivery timing, and other times you use something different. I mean, I think it just has to be an open question, which is, yes.
Maybe related to that, in the other part of, I forgot the name of the Oli, not the PO, but the CO.
Oh, sorry, the DC, the dorsal cap of curve.
Thank you. So you said that there are no subpressure oscillations there.
Well, okay, just a minute, what am I trying to do here? Let's go back to the...
Oh, here.
Okay, here it is, yes.
You're saying that there are not, in the DCK, there are no subpressure oscillations.
Or at least they're much they're much weaker.
Yeah. But do we know about the firing rate of the climbing fibers in those areas? Are they still 1 Hertz?
We do know about the climbing fibers projecting to the flocculus. Yes, that it averages 1 Hertz.
So,
but I don't know. Do you know?
It's the same.
It's the same.
Roughly the same.
So if it is not the adaptive filter, but it's really the online control of movement, then it's hard to explain how such slow firing is good to the movement, to the...
Yeah.
Well, you always have a problem with the low firing.
I have a problem, but I run fast, I don't know.
But-
Somebody walks slow, then...
But you have a million Oliver neurons. A million Oliver neuron firing at 1 second, how much is that?
A million seconds.
Thank you, you're the best.
You're welcome.
Even time cannot understand. No, I think that the fact that it's firing very slow, it's not, I don't see it as a problem.
But why should fire slow? Just because to
Saving energy.
million climbing fire. So the Purkinje said you don't say million, you say Purkinje fire like hell. And so there must be some rationale for that. The vast difference in the single neuron firing rate should explain that.
No, I mean, people were worried about this problem long ago,
Okay, very
and
good.
that's kind of the idea of teaching that the arrow signal came for that.
That's
It
fine.
makes sense, you substitute synaptic points with firing rates. That's what you said.
The argument, I think, as I said, it always was that it seemed a bit kind of eerie almost, that an adaptive filter, you've got to somehow marry what look like two opposite requirements. One is it's got to be extremely powerful, because it must alter all the synapses. And the other is you don't want to influence, you don't want to know about this in your output. And so it seemed to be the fact that the average firing rate was 1 hertz, whereas perhaps the output firing rate, the simple spikes, is about 50. It just seemed to be a tremendous
No, I
solution to
agree.
that
It
problem.
seems to be consistent with adaptive filter. It's mysterious to me how it will be consistent, how it's consistent with other sensory modes, with a more motor control cut. This is different from adaptive filter.
Mm-hmm. Mm-hmm.